Hello, everybody. This is actually a class at the University of Arizona, uh, part of a new undergraduate minor in consciousness studies. If there are any interested students that have tuned in for this, if you want more information about it, you can write to me, Tom Bever, or you can write to Laura Owen, who is in charge in the SBS Dean's Office. This is being offered in association with the Center for Consciousness Studies and the new Center for Astrobiology. As I say, it's part of a class, actually, so students will be involved in some of the discussion. But we decided to open it to the larger audience, both within the university and in community, because of its general interest, because of the three particular people who will be leading the discussion that is actually part of a class, but has a lot of general interest in it, as you shall see. I'll start off with a few remarks, somewhat casually, and then each of the visitors will make a presentation, which will be followed by some general discussion between them and also an opportunity for students who are in the class to actually ask questions or make comments. So here we go. Here are my initial remarks. I'm going to start with a short anecdote to illustrate some of the contrast between intuition about the world and science. It goes back to a trip that I made with my son, who was about age 11 or 12 at the time, to see the great United States. And we were driving along and we were discussing theories of the heavens, of the cosmos, of the solar system, with my fairly primitive knowledge of it all, but enough to describe it as we all sort of intuitively think about it. And at the end of the discussion, he got out of the car. We were outside a tent lot somewhere. He got out of the car, looked at the ground, and said, "No, Daddy, it's flat. It's really flat." And what that symbolizes or encapsulates is an intuitive notion of the world that we all believe must be true, when science of one sort or another tells us that it isn't, and that we have to think about it perhaps in an entirely different physical way or a much more abstract way. And that's what we're going to face in some of the discussions tonight, that kind of contrast. So to shift to some well-known examples of this, we look back at, for example, the history of our beliefs about planetary motion and the solar system, and we imagine, when we just think about it casually, that it all came together to a current day thinking about it all very quickly. I mean, we tend to smush together in our ideas Galileo, Copernicus, and Newton, like they were all together and then conspiring in some room to straighten things out. Well, of course, it was much more spread out in time. But the important aspect of it for our consideration today is how it ended up. It started with a view of the world which was, we would think in today's terms, quite mechanistic, like the world is a giant machine and everything is physically connected to everything else in a way that my son would understand at that age, and that anybody would tend to accept as a model, even though you can't see some of the things that actually physically connect the objects. Well, with the advent of Newton's thought, an examination of the prior ideas of Galileo and Copernicus, he puzzled and actually came up with a much more elegant solution, namely the equation embodied in part in the equation we know today in so many different forms, force equals mass times acceleration. And that seemed to explain so much once it's properly configured in relation to the planetary motion, mated with circles and so on, seemed to explain so much that it obviously had to be believed. But for a long time, Newton made a point of not really believing it. In fact, he said something to the effect that no rational man would accept this view, namely the view of a non-mechanistic universe, action at a distance, being the culprit that is inconsistent with the notion of a mechanical universe with connections between everything. But there it was, the power of the equations eventually in relation to other observations that continually enrich the evidence for that way of thinking about it. So we're left with a theory that nobody has natural intuitions about, but it's a theory that at this point is so generally accepted that we do take it for granted. Well, today, the real question is about how this might apply to our understanding of consciousness. One of the reasons that we started the undergraduate program in consciousness studies was my intuition that this is a good time, a ripe time, for an eclectic approach at the undergraduate level to bring together, at the level that undergraduates can appreciate, various streams of new kinds of knowledge and theory that have emerged in the last decade or two. And that, in a sense, 30 years from now, 50 years from now, centuries from now, who knows, we may look back at this time as a period similar to that, that we tend to compress as the period joining Galileo, Copernicus, and Newton, that there's a kind of crucible of different approaches to the problem of what consciousness is and how to study it and what its history is, that are coming together and that are instantiated by the speakers today. So the people today, the speakers today, are going to engage in a discussion that is wrapped around a particular theory of what consciousness is. You'll see that it's kind of abstract in the same sense that Newton's hypothesis about gravity was abstract. It didn't accord directly with intuition, but did such a great job of predicting so many facts it became accepted. The question is whether today's theory that they'll be talking about it has attracted some empirical support, and of course the question is whether it will ultimately become the received word. We don't know. But this meeting encapsulates at least approaches to it that represent the state of the art today, with three brilliant people, each in a separate central discipline that converges on the question. So in particular, the theory that is the common theme you'll hear about first from Stuart Hameroff, sitting in the center over there, who will introduce the notion of a theory that is based on quantum theory and how certain kinds of quantum effects apply to very tiny parts inside of neurons in particular to engage a particular kind of process which one could interpret as consciousness. And that's embedded in Stuart's theory that is what consciousness is at a physical level. So there are three approaches today in general. One will be Stuart's exegesis of that particular theory, studying the implementation of quantum effects and how consciousness arises. Anirban Bandyopadhyay will emphasize the physics of this, some of the basic physics of that, and will talk about certain kinds of evidence that introduce magnificent heavy use of fantastic microscopic type tools, and also combined with the deep mathematical analysis of how to interpret what those tools are showing. Dante Lauretta will turn to the latest attempts, very recent, to explore the earliest stages of the universe. and how they may provide, how those earliest stages may actually provide some of the initial physical opportunity for consciousness to be enacted. The three together offer us an astounding hypothesis, really astounding. First, it starts with the real support of the theory and description of it. The second is an analysis in part of its contemporary basis with new kinds of data and analyses. And the third is now bringing in some historical data. By historical, I mean data that go back in time, we hope as close as possible to the founding of the initial creation or founding of the universe. The astounding hypothesis that we will see is that consciousness has been present in the earliest stage of the universe and has played a formative role in the evolution of certain aspects of the universe. Certainly part of the astounding hypothesis is that consciousness has played a causal role in the emergence and the increasing complexity of mechanisms that convey and have consciousness available to them, like us. So the first speaker is Stuart.
Thank you, Tom. Hello, everybody. It's good to be here with Tom and my two friends and colleagues, Anirban and Dante. And hello to all of you. Thanks for tuning in. My topic is which came first, consciousness or life? If we can see the first slide, please. So which came first, life or consciousness? And what you see in the background are molecules floating in space. These are polyaromatic hydrocarbons, and Dante will turn to them later, and that's kind of the punchline because that's where we're going. Next slide, please. So most would say that life came first and consciousness evolved later, perhaps only recently with language and tool making. Others, for example, panpsychists, Eastern spiritualists, and some quantum physicists say consciousness came first and has been here all along. If this is so, could conscious feelings have sparked the origin of life and driven its evolution? This is what we are going to argue for. Next, please. So let's first try to define life. Many people say, well, we solved life, now let's move on to consciousness. We haven't really solved life. We really don't know what it is. There are several approaches. For example, the functional approach put forth by Lynn Margulis Sagan, describing self-organization, metabolism, growth, adaptation, response to stimuli, replication, evolution. These are functions of what life does, not what it is. A second category would be complexity, nonlinear dynamics, scale invariant logic, information flow, coherently pumped metabolism, emergence, a nonlinear dynamics, self-organized criticality. There are many of such categories, but inanimate systems have these. They are not unique to life, and it's not clear that they cause or responsible for life. They're more descriptors. Next slide, please. Then there's vitalism. In the 19th century, a group of people described a unifying energy field, a life force, or elan vital, pervading living systems. But these were based either on electromagnetism or on unknown forces outside science. With molecular biology, the discovery of DNA, and so forth, biology got tired of vitalism, and it became taboo. And the idea of a life force was shunned. And then in 1935, Erwin Schrödinger, a famous quantum physicist, wrote a book called What is Life?, where he suggested that life's unitary oneness derived from quantum coherence, quantum effects, and that memory was stored in aperiodic crystal lattices. This solved a lot of problems, but it was said that biology is too warm, wet, and noisy for functional effects in quantum systems and easily perturbed by thermal decoherence. Or is it? We'll come back to that point. Next slide, please. Let's next try to answer what is consciousness. We didn't answer it for life, but let's look at consciousness. And the definition can be awareness, experience, what it is like to be, first-person view, self, qualia. These are just words. You know what consciousness is if you're conscious, and I assume you are. I know what it is, but it's really hard to even define it. The approaches to explaining consciousness, probably the most popular, would be complex computation among simple neurons. The brain is a computer and consciousness is an output. Many problems with this. Integrated information, it doesn't matter about neurons, it's just information which gets processed in a certain way and consciousness emerges. That's also very problematic. Predictive coding says that you expect something and you compare the present to what's coming in, and the difference is what goes into consciousness, some novelty and so forth. This is actually a very useful function and probably happens at many levels in the brain and in the nervous system. So as a complete explanation or definition, it's not appropriate, but it is very useful, perhaps the most useful one among the prevalent theories. And then finally, that consciousness is a fundamental feature of reality. For example, Eastern spiritual approaches, David Bohm's quantum potential, and objective reduction, a self-collapse of the quantum wave function as a process in fundamental space-time geometry, put forth by Roger Penrose, with whom I've worked to develop a theory of this based in structures called microtubules. Next slide, please. So most view the brain as a complex computer of simple neurons. Neuron firing equals 1 bit. So here we have a neural network and with hidden layers, and information comes in from the left, percolates through these adjustable connections, which are analogous to synapses, to an output. Next, please. Consciousness is theorized to emerge from higher order network effects. Next, please. This idea is an insult to neurons. Neurons are shown here as empty, cartoon-like, algorithmic input-output devices, and simple cells are much more interesting and capable than this. Next slide, please. For example, a single-cell paramecium is an organism. It can swim around, it can find food, avoid predators, avoid obstacles, as we see here, and have sex with a mate. And Bing is meant to imply consciousness, suggesting that maybe even single-cell paramecia may be conscious. And this is actually a valid point, because what else motivates them to do what they do? To avoid getting eaten, to find a mate, to find foods. It could be that they have primitive consciousness and they're trying to feel good. It makes them, that actually makes sense if there were a mechanism at that level to explain it. That's the problem. There hasn't been until fairly recently. Next, please. The paramecium do what they do by their cilia, which are the projections that stick out, made of structures called microtubules, and neurons are chock full of microtubules, as we see from this cover, the journal Neuroscience. The long filamentous processes are structures called microtubules that make up the cytoskeleton of neurons and other cells, but are also, we think, the nervous system of the cell, of each cell. Next, please. And if these microtubules fall apart, we get Alzheimer's disease. And while most of the attention and research goes toward the amyloid plaques, which are outside the neurons, it's actually the microtubules and the tau proteins falling off of them inside the neurons, which cause them to disintegrate, lose synapses, lose neurons, brain atrophy, lose memory. And it's the microtubules that are really the culprits in Alzheimer's disease. Next, please. Now, back in the 70s and 80s, I, for one, got interested in microtubules as information processing devices and was looking at systems called cellular automata, like the game of life, a simple computer-like game based on a simple grid, orthogonal grid, where each state could be dead or alive, depending on the number of neighbor states alive in the previous generation. And with these very simple rules, you can generate patterns that move through the lattice and can perform functions, including solving algebraic equations and nonlinear partial differential equations and so forth. And we compared that to what might be going on in microtubules, which you see in the lower two columns in microtubule automata. And the particular hexagonal lattice in the microtubules provide with the cylindrical geometry and Fibonacci geometry gave very interesting patterns which wrapped around and gave rise to interference and also capable of processing information. And each microtubule is a lattice polymer of these proteins tubulin. And so we were likening the tubulin states to bits as opposed to a whole neuron to a bit. Next, please. The neighbor interactions occur at a specific clocking time, a frequency. So to make this model work, we needed some kind of coherence, some kind of coherent oscillation of all of the components oscillating coherently together so they can interact and process information. Next, please. There was a theory, and still is a theory, by a man named Herbert Froelich, who was a researcher in superconductivity who turned to biology, and he put forth the idea that there was quantum coherence at ambient temperatures in structures in biology, including membrane proteins and microtubules. I had the good fortune to meet him in the 1980s and told him about microtubules, and he agreed that they were good candidates, and he invited me to write a chapter in his book. And he basically said that coupled dipoles in nonpolar regions of pi electron resonance, which I'll talk about briefly in a minute, in geometric arrays and common voltage would oscillate coherently in frequencies including terahertz, gigahertz, megahertz, and kilohertz in these geometrical lattices, condensing to common modes and creating a quantum effect, a common quantum effect, something like a laser. Next, please. And so we developed models and theories of the microtubules inside neurons. And you can see here on the left, there's a synapse with an axon on the far left, a dendrite with the microtubules being broken and interrupted for reasons that are unclear. And on the right, you can see patterns moving through the microtubules. And these could be classical computing on the left or even quantum computing, as I developed with Sir Roger Penrose later on. Next, please. Now, this was a great theory. It required some kind of evidence, and fortunately, that was provided in terms of in terms of coherence, collective oscillations in microtubules by my colleague and friend Anirban Bandyopadhyay, who, in a series of papers from 2013 to 2020, studied microtubules at 3 different scales. In the top, you see neurons in a neural network. And in the middle, you see individual microtubules, and below you see rows of tubulin dimers, the components. So 3 different scales. And if you apply a voltage to a microtubule, it's a good insulator. There's no current flow. But if you apply a voltage that oscillates or an alternating current, you'll find certain frequencies where there's a significant conductance, a huge increase in conductance. And that's what's mapped in the middle set of images. And what Anirban found was that the conductance patterns repeated every 3 orders of magnitude, just like Froelich suggested, terahertz, gigahertz, megahertz, kilohertz. And the conductance at each level had a particular pattern, a triplet of triplets. So 3 peaks, and each peak had 3 peaks. This triplet of triplets that Anirban found, I think, is going to be very, very important in understanding many things, including consciousness. Next, please. So the point is microtubules have coherent cell-similar resonance patterns. Next, please. And these triplet of triplet patterns repeat every 3 orders of magnitude. Next, please. And there's Anirban, who's also sitting next to me. Next, please. As an illustration of this, you can also detect these triplets from the scalp. And here's Hide Sagusa holding a probe to his head. And on the monitor, you can see a triplet. I don't know if the video can run, but if he takes it away from his head, it goes flat like that. And if he puts it back, you can see the triplet. It's a little subtle, but you see the flat line when nothing's there, and 3 peaks. Okay. And within those peaks, presumably within each of those peaks are 3 peaks within each one, triplet of triplets. Next, please. And these are in megahertz. This gives rise to the idea of a scale invariant hierarchy. Many people have suggested this for the brain, starting from the neuron, going upward into networks and networks in the EEG. So kind of coherent phase relationship between EEG patterns, frequencies. But it also should go inward, deeper, faster inside the neuron. So on the left, we have a pyramidal neuron, which will oscillate in the EEG frequencies. And as we move to the right, we have a network of microtubules that has oscillations in kilohertz. And then moving further, a single microtubule in megahertz, rows of tubulins in gigahertz, individual tubular in terahertz, and then the dipole oscillations in petahertz. And this level, hundreds of terahertz or petahertz, are where anesthesia acts to block these dipole oscillations. So this is the level we have to go to, at the level where anesthesia acts to selectively block consciousness while not affecting anything else. The brain is quite active under anesthesia. The only thing that's eliminated is consciousness. And then further down into petahertz, and then all the way down to the Planck scale, where Roger Penrose's ideas come in of space-time separations. We'll come back to that point. Next, please. So that leads to the question of quantum superposition, because as we move along that spectrum, we eventually get into the quantum world. We get into smaller, faster, deeper activities that are quantum. And the world seems to be divided into two realms: the classical realm that we're familiar with at our large scales, and smaller, faster levels where things can be in multiple locations at once. For example, in quantum superposition, a particle can exist as waves of multiple possibilities, or as a particle in a definite state and location. Classical. So in the front, we see a cesium atom in one location, but in the back, we see the same atom as a wave. Next, please. It seems that that a particle can exist as a wave of possibilities, but the very act of measurement or conscious observation seems to cause the wave function to collapse to particles in definite states. This is called the measurement problem in quantum mechanics, and Schrodinger's cat was intended to poke fun at it, but it's still an enigma today. Next, please. Now, how can we explain this, and how can we explain superposition in the first place? How can things possibly be in more than one state or location at the same time? And Roger Penrose brought in general relativity to explain this, pointing out that mass, shown here for large planets and celestial bodies, are curvatures equivalent to curvatures in space-time, in fundamental space-time geometry, Einstein's space-time continuum, or metric. Next slide, please. Roger applied this to tiny particles and tiny curvatures for example, using these two-dimensional sheets to illustrate. So on the left, above and below, we have a particle in two different places with its attendant space-time curvature oscillating back and forth. And on the right, we have a superposition of this one particle in two different places with separation in space-time geometry. Next, please. So this is a good way to envision the measurement problem. If separation were to continue, each particle would have its own universe. And this is actually the main point of what's called the many worlds hypothesis, that there is no collapse, that each possibility goes off to form its own world, many worlds. And you can envision this happening by space-time separating into two universes. Next, please. Another possibility is that conscious observation causes quantum state reduction, and this was put forth by quantum pioneers von Neumann, Wigner, more recently Stapp, Chalmers, and McQueen, that consciousness causes collapse of the wave function. We see Bing in the observer's head, and when the observation affects the superposition so that one possibility terminates and the other one continues. So the wave function has collapsed, and one possibility is selected. Next, please. Roger Penrose had a different idea, that these separations were unstable. And after a time t, given by a very simple version of the uncertainty principle, t equals h bar over e sub g, it would one possibility would collapse spontaneously due to separations in the universe being unstable, and cause a moment of conscious, of proto-conscious awareness or experience, Bing. So the idea, instead of consciousness causing collapse, putting consciousness outside science, was that collapse occurred spontaneously due to an intrinsic property of space-time geometry. So rather than consciousness causing collapse, collapse caused consciousness, or is equivalent to consciousness. Next, please. And this would be ubiquitous in the random microenvironment. Next, please. And but would need to be orchestrated to have the kind of consciousness we have ourselves today. So we'll come back to that point, but let's go back to the question of which came first, life or consciousness. And here we see a history of evolution from the early Earth to modern day, and with some landmarks in evolution. Next, please. So where did consciousness appear? Bing. Where's the Bing? Next, please. So one possibility that many people believe is that it's fairly recent, with if not humans, our fairly recent ancestors with development of language and tool making. Some people would say that prior to that there was no consciousness. How they explain all that behavior, purposeful behavior, all those creatures doing stuff to survive, is a good question, which is why I think something else is needed. So I don't think that's the case. Next, please. Another possibility is that consciousness arose at the time of the Cambrian evolutionary explosion, a fairly brief 10 million years, 540 million years ago, during which all of the animal phyla developed. And you see, as it's projected down below, some of the organisms that were present in the Cambrian evolutionary explosion, including something that looks like an actinospherium, something that looks like a nematode or something, and a sectorian. And if we look at some of those organisms that exist today, including the actinospherium, next, please. This is that same organism on the top, and it has these axonemes sticking out like spikes, which if you cross-section them, you find a double helical array of microtubules in cross-section. So these things are completely made of microtubules sticking out with membrane in this double spiral array. And if you administer anesthesia, they depolymerize. And in fact, this was once a theory of anesthesia put forth by John Nunn in 1968, that anesthesia worked by depolymerizing microtubules in our brains. Fortunately, it takes a lot more anesthesia to depolymerize than to affect them. And so we're not depolymerizing anybody's microtubules, except maybe in excessive doses, which results in postoperative cognitive dysfunction and perhaps Alzheimer's disease. Below, we have a sectorian from a modern day sectorian that shows that it is also made of microtubules. And we see one microtubule on the right. So the point is that these urchins and simple organisms had microtubules and may have had something to do with the proliferation of all the phyla and this tremendous explosion in evolution in a brief period of time, as found in the Cambrian shale up in Canada by Stephen Jay Gould. Next, please. So going back to Roger's idea, he needed a quantum device in the brain which could orchestrate quantum information, halt or terminate by his objective reduction, connecting to space-time and possible platonic values and qualia, and to regulate functional neuronal and synaptic activities. And I suggested to him back in the early 90s that microtubules were what he needed. And next slide, please. So we developed a theory based on tubulin, and I'll come back to the evolutionary story in a minute, but this is a tubulin with many aromatic rings, and the red spheres are where anesthesia acts. And these aromatic rings are where quantum effects can occur inside biology and be unaffected by the supposed warm, wet, and noisy environment, because they're in quantum-friendly regions inside tubulin proteins due to these aromatic rings, the basis for organic chemistry, polyaromatic molecules, which we'll hear more about shortly. Next, please. So aromatic rings start with benzene, and they have 3 extra electrons, which form these electron clouds above and below the ring. And these electron clouds are more or less quantum objects of superposition. And they support quantum effects, including dipole oscillations, excitons, spin transfer, phonons, fluorescence, phosphorescence, subradiance, and superradiance, and are found inside proteins and inside many biomolecules in a quantum-friendly environment. So the brain is not actually too warm, wet, and noisy inside proteins, and particularly microtubules. Next, please. So these structures give rise to fluorescence, where if you excite them with an ultraviolet photon, they reach an excited state that percolates along and then drops down fluorescence, giving lower energy light. And this is depending on the propagation time and how long and distance, this can be a quantum effect. Next, please. So here's the simplest form of 2 benzene rings. They attract and they induce dipoles in the other, and they oscillate in terahertz. And this is the origin of oscillations in petahertz and terahertz inside molecules. Next, please. And it should also be noted that psychoactive molecules, including dopamine and serotonin, and the psychedelics, LSD, DMT, and psilocybin, are composed of these aromatic rings, or largely composed of these aromatic rings. And so this is a further hint that these have something to do with consciousness. Next, please. It could be as simple as the arrangements between aromatic rings giving rise to feelings, such as there's two stable arrangements, the T-shaped or the offset parallel displaced, and maybe they correspond with different qualia. So this would be a simplistic approach, but feelings have to come from somewhere, and it could be that they come from collapsing to particular configurations of aromatic rings. Next, please. And here's a kind of, excuse me, like a quantum bit based on two rings that can collapse to either happy or sad. So this is a general suggestion that might be true. Consciousness and feelings have to come from somewhere. Next, please. And we did a study published earlier this year where we studied a microtubule and used ultraviolet light and found it propagated an exciton and then gave rise to emission. And the propagation time and distance was too far and long for classical effects. So this was a quantum effect. And then we showed that it was inhibited by anesthesia, two different types of anesthesia. And we also realized that microtubules are efficient light harvesters and may actually deal with photons in information processing and consciousness. Next, please. So here's the tubulin again with its 86 aromatic rings, which is a huge amount for this size protein. And we developed a pathway, a dipole pathway that could oscillate through tubulin. Next, please. And this became a topological qubit that wrapped around the microtubule and could oscillate back and forth. And these oscillations would lead to superposition states, which would collapse for a conscious moment, an orchestrated moment of consciousness. Next, please. So here's where the superpositions reach a threshold for Bing, which is meant to mean a conscious moment. Next, please. And so that's our theory of consciousness. And if we go back to the evolutionary story and go even earlier to the Bing being at the origin of the eukaryotic cell, this is supposedly when prokaryotic cells were invaded by spirochetes, bringing flagella, which contained microtubules, although this theory is now questionable. And we really don't know where microtubules came from in the big scheme of things. Next, please. It's the spirochetes invading prokaryotes is analogous to the sperm invading the egg, which triggers off life, including a microtubule mitosis. Next, please. And the flagella in the tail of the sperm and in the spirochetes are made of microtubules, or are dependent on microtubules. They're the structural component of it. Next, please. And so if we go even earlier, we come to first life. And I think this is where consciousness actually is found, already there. Next, please. So let's talk about that. Life on Earth apparently began in a primordial soup proposed in the 1920s by Operin and Haldane, a simmering oily mix from which biomolecules emerged 3 to 4 billion years ago. Next, please. In the 1950s, Stanley Miller and Harold Urey simulated a primordial soup with nutrients, an oily mix, and lightning sparks for lightning. Next, please. And they found amphipathic biomolecules with the organic aromatic rings with the pi electron resonance clouds we talked about earlier on one end and polar water-soluble tails on the other. Next, please. In protein folding, amphipathic pi resonance clouds on aromatic amino acids attract by transient-induced dipoles and extend their polar ends outward. This is how micelles form in the primordial soup, according to Operin and Haldane. And on the right, you see what could be a simple micelle with 3 rings in the middle and the tail sticking out. Next, please. So back in the primordial soup, next slide, please. We can imagine that a primitive micelle with its aromatic rings reached a critical E sub G and had a conscious or proto-conscious moment at time T equals H bar over ECG, and a bing, or maybe with a small letter bing, with primitive proto-conscious feelings of primitive events, bing, occurring, emanating from the level of space-time geometry. Next, please. These would be random in the microenvironment, but some would be positive, feeling good. Next, please. Perhaps. And this, therefore, could have, next, please, led to self-organization of those micelles and the aromatic rings in the micelles to optimize pleasure. So the question we're posing is, with pleasure as a feedback fitness function, ordering pi resonance groups, did life then evolve to orchestrate and optimize OR-mediated pleasure? And we call this the quantum pleasure principle, borrowing from Freud's Beyond the Pleasure principle. Next, please. So Darwinian evolution is a pillar of modern science, but the notion that life evolved behavior to promote gene survival, the selfish gene hypothesis, is merely an assumption. It doesn't make sense. Excuse me. Behavior in a laboratory animal, in animals in general, in people, in us, even today, our behavior is driven by reward. We may delay our gratification if we're mature adults, but it basically revolves around pleasure, avoiding displeasure, and feeling good one way or the other. There were no genes in the primordial soup. It took a long time to get to genes, and certainly no brains. And evolutionary theory ignores consciousness and feelings because they don't have a mechanism for it. So they make up these stories to get away without it. Next, please. So did feelings drive evolution? That's the question. Next, please. And this goes back to our spatial hierarchy, where you can see the triplets of triplets at every level, including, we propose, down to the Planck scale. And we think this is something that may be tying things together. Next, please. So back in the primordial soup, anywhere else? Next, please. Polyaromatic hydrocarbons are produced by stars and float in space, in interstellar space, in interstellar dust, atmospheres of exoplanets, found on the windshield of the International Space Station, in meteorites and asteroids. And Dante will be telling us much more about that. And next slide, please. So I'll conclude by saying that life may be quantum in nature, its essence in decoherence-free subspaces, what I call the Meyer-Overton quantum underground, the Meyer-Overton telling us where anesthesia acts, which is an adiabatic decoherence-free subspace protecting quantum effects. Regions of aromatic rings and nonpolar biomolecules sequester, avoid water and polarity, enabling limited degrees of freedom and quantum mechanism to be isolated adiabatically in lattices like microtubules to resonate and interfere. Consciousness in the brain is likely to occur in a scale invariant hierarchy in mixed polarity microtubules, in cortical layer 5 pyramidal dendrites, and soma, more like a quantum orchestra than a computer. And finally, proto-consciousness likely preceded life, prompting its origin and driving evolution. Thank you.
Thank you. As I said, it's an astounding hypothesis. So let's start, if students have some questions or comments first. So let's start with that. If there are any students, please, I guess, raise your hand on the screen as you would ordinarily, and ask Stuart whatever you want.
I can't read that.
No, there's one in the chat there.
Can somebody read the chat?
Oh,
It went away.
consciousness and its components are universal, why do we see higher levels of consciousness on Earth? Why don't we see conscious organisms on other planets? What is unique about Earth?
Well, we don't know. We haven't looked, or we haven't looked properly. It could be that they are higher frequency. It could be that they're not embodied. It could be they're pure energy or orchestrated information. It could be that they're distant and can't reach us, or we can't reach them, and maybe they just don't want to mess with us because we're not worth it. We don't know, but we're going to find out. We hope to find out.
Well, be careful what you wish for.
I can also address that just as for the astrobiology side, that's exactly what we're doing, is looking for life elsewhere in the solar system. We think there are several good targets, the subsurface of Mars being top priority, but also there are oceans in the icy satellites of Jupiter and Saturn. We see them spewing geysers out into space, and we're targeting those for extraterrestrial life forms as well. And of course, now we have thousands of extrasolar planets, even within our vicinity of the Milky Way galaxy, and we are looking for abodes for life there as well. So I think, as Stuart said, it's way too early. We've just gotten started looking for life, and we don't understand the exact environmental conditions that are required for it to originate and also to evolve.
He should know. He's the astrobiologist.
Yeah. So
We're all astrobiologists.
are there other questions? I mean, please.
Do you think that consciousness is something that we tune into like a radio? We all have just different mechanisms and perceive it differently.
I get asked that a lot, and I would say it's a hybrid. If you believe the brain is a computer, then definitely no, the brain generates everything that comes into consciousness. Some people take the opposite view, that the brain is just a conduit and our radio receiver, our TV set, receiving inputs, and just that's experience. I think it's a mixture. I think we generate content through the course of our lives, our memories, and the evolution and so forth. But we are influenced by something out there, but it's much more subtle than a TV set. I think it's actually intrinsic to the universe, and what Roger Penrose calls platonic values, something embedded in the geometry of space-time that our thoughts somehow resonate or get in tune with, but more subtly than us being just pure TV sets.
I have a question from Jared Lovemark. Do you want to speak up, Jared?
Yes, thank you. Regarding your quantum consciousness involving microtubules, is there any idea of like what the information form is that microtubules might be processing, or is it just so far a theory of what in the brain might be making the computation process?
I've evolved on that. I used to think that it was just bit states of tubulence, but now I think it's a topological quantum qubit having something to do with the triplet of triplets, because that keeps coming up over and over again, and it's becoming harder and harder to ignore. So Anirban's going to talk about that. So something to do with triplets of triplets and a topological qubit in some kind of self-collapsing quantum computer.
Thank you.
Okay, I think we should move on. And I'm just going to make a short interlude to introduce everybody a little bit more. So Stuart, who just spoke, if you couldn't detect it from his focus, started his academic life to some extent as a doctor, became interested in anesthesia and anesthesiology, and then became fascinated with the question of how we make people unconscious as necessary for surgical procedures. And very early on became fascinated with microtubules, which has dominated his attention, and then radiating from there the theories that he just spoke about for 30 or 40 years, as far as I can figure out, or possibly a bit more. He has been the director of the University Center for Consciousness Studies for quite a while, and made a lot of contributions to the field in that way, while continually working as an anesthesiologist in the hospital setting, which he just recently retired from to move into a position as a research scientist. at the university. The next speaker will be Anirban, who is an absolute wizard, combining physics with powerful mathematics and ingenuity experimentally. So among other things, he invented and developed the use of extremely tiny electrodes, is the only way I can describe it, although I'm sure it has a more sophisticated description, which allow recording events inside neurons, which is a tremendous advance just by itself. And you'll see in his talk other wizardry and powerful mathematics applied to images that he has developed methods to achieve, and also borrowing other technologies to integrate with his analysis of some of the basic phenomena that Stuart was talking about that can underlie consciousness. Dante, way over to the left of my left, is recently the director, and when I say recently, that means at least 10 years, I think, or maybe a bit more, of the major project, Sirius, to collect bits and pieces of space stuff, rocks and whatnot, whatever there is, from an asteroid in order to be able to perform modern style analyses on the result that will inform the kind of theory that Stuart is talking about as one of the few theories that actually has testability of that kind. Dante has just started a new institute at the university, I guess a center for astrobiology, which will be focusing on these sorts of problems as his newest adventure. And I think that we will now turn to Anirban.
Yeah, so David, can you load Consciousness 2 file? Okay, so thank you, Tom, for introducing me, and I will briefly introduce some features of triplets that you have learned from Stuart moments ago. And to the left, you can see that I'm playing with a triplet, and they are born together. And I have put 3 letters, R, S, and T. The gentleman with T, he always hugs others, loves everybody. That is the bonding. So instead of bits and qubits, we found microtubules, tubules, and then neurons. We do not want to look at them as just like a bits like computer chips or transistors. We found that clock-like vibrations. Triplet means 3. We say triplet of triplet means 3, 3, 3. That is 9. There are 9 different clocks together. So they try to bind them together. So that is T. It is from Tamas, the ancient word. In the middle, the kid is S. He is always philosophical and always used to teach me how to live life properly and how to handle difficult situations to me. And that is S. We call it Sattva. And in my left side, he is always jumping and dancing. And I mean, you can't find even a fraction of a second as a stop. So that is Rajas. So that is the third property of the clocks that they have. So the clocks are say 3 clocks forming a triangle. Another clock comes and then they become a rectangle. So that is called transformation. So transformation, then projecting the geometry of clocks to the infinity, getting the feedback, and binding. These are the 3 fundamental properties, and I experienced them personally in my life. In this picture was taken in Boston. To your right, you can see that there is a small pie chart, and this pie chart actually fascinates me a lot. This is in our body, 62% water. So imagine I have this bottle. So 300 ml is water. And then I will put say 60 ml of proteins, 60 grams of proteins, and say 60 ml of oil. and say 30 grams of salt of different types, minerals. And then I will shake it very well, and that is me. Okay, and that becomes conscious. The question is how? And that's a mystery that we have. And I mean, often in the dream, I feel like water wanted to be conscious. That's why it made us. And we all stupid guys are looking into proteins and others and trying to solve this problem. Maybe one day we all will be dead and then somebody would figure out that, I mean, it was just a mystery of water. Next slide, David. Okay, so in the bottom part of this slide, you see there are two images side by side. This is a very famous, it's called millennium simulation. So what the scientists did is they took the particles and then assembled them together and tried to simulate. And at a particular phase, to the left, you can see it is the galaxy, and to the right, it's a neural network in the brain. So you can understand that from the smallest to the largest, there is a relationship of symmetry. A moment ago, you got a very good presentation from Stuart, who linked smallest to the largest with triplet of triplet. So 2014, we published it as a resonance chain. And so, I mean, is the resonance chain starts from the elementary particles? Does it go to the outer galaxies, even beyond? And the whole universe could be a whole resonance chain, and we are a subset of it. And we are part emulating the whole, and that's why we are conscious. So these kind of simulations inspire us to think about consciousness very differently. David, can we go to the next slide? Okay, to the right, there is a video. Can you play? If you just bring the cursor to the bottom, then the arrow button will be just a little below. The arrow button will be there. Cursor should come to the bottom. No, not there. To the right, to the right, to the right. Yes. No, I don't think so. The cursor is over there. If cursor comes down little below, you should see an arrow. Okay. You try to click there. Do you see anything running in the middle of the video? Okay, it goes to the next slide. Okay, fine. So I will not be able to show any videos, it seems. That's fine. So here in this video, I try to show that from the time of dinosaurs and to now, if we look at the evolution of the brain, then we will find that we have grown the number of neurons, and a very particular part, you see the big two spheres, they will expand and eat up the whole volume. So that gives us an idea that we should always try to find consciousness in the neurons because large number of neurons are there. But there are people who are living very nice, very well, very conscious, and due to hydrocephalus, they have empty brain. I mean, most of the neurons are gone. Very small neurons are left. Can you go to the next slide? Okay, so what are neurons and what are the structures? Where does the microtubule sit? So if we go inside the brain, so it is now believed that at the top part of our brain, just below the skull, there is a thin layer of a few millimeters. And in those regions, there are hexagonal close packing, crystalline packing of cortical columns, around 200,000. And there are 47 these regions, functional regions, which gives us the different cognitive, perceptive, and conscious experiences. And those cortical columns are made of neurons, the cell branching like trees that you can see here. And there are synaptic junctions or ion channels, which are very, very small, tiny few angstrom, so that ions can pass through very fast. And it is believed that those ions are doing everything. But we are challenging this concept. And if you go to the bottom part of the slide, then you can see that there is a string-like structures. Those are the microtubules. And microtubules are the string. And in most of our textbook, when you study, they show that membrane is there and inside it's filled with liquid. And only in the very higher studies, you find that it is actually packed and very strong structures are there. So can you go to the next slide? Okay, but we try to see biological system from a very different perspective. If you look at the atomic structures, they vibrate in a particular time, and if you go to the dipoles, they vibrate from picosecond to nanoseconds. Large dipoles like microtubules, they vibrate in microseconds, and there are many different structures, different biological events that are happening at all time domain. I mean, even if we think that we live for 100 years, it's a 10 to the power 9 Hz oscillation, just one big. So could it be possible that all the clocks are integrated together and a very structured information is there? Because if we want to understand consciousness, four questions we need to answer. How the information looks like, what is the information, and what is the elementary device that processes information, and how information is integrated, and finally, how we become self-aware. So when we go for, instead of past, present, future, consider time as a clock and take a sphere and then put a lot of sphere together, then our perspective to understand consciousness or biological system is fundamentally very different. Can you go to the next slide? Okay, so one particular concept that we should always know, and that is Turing machine. Why we should know? During the World War, when the world was in a very different place, a unique concept came called Turing machine, and it said, it's philosophically very strong. It said that whatever be the information in the universe can be written on a linear tape with 010101, yes or no, yes or no. How true is it? We don't know. But even after the war, when all killing machine, our quest for killing machine were over, we did not go back to nature and try to find out what is the structure of information. We kept with that, and it's still there. So 99.99% conscious theories, I mean theories of consciousness, they make formulations and propositions based on this particular machine that you are seeing. So brain is almost like a computer. Some of the proposals like integrated information theory and others, they say non-Turing machine, but they don't understand what is Turing machine at all, because all their formulations, if you see, are mathematical formulation. If you can define any state, it cannot be a non-Turing system. It will be Turing computable. The most fundamental argument was made by Roger Penrose in 1989, and he continued to work on that. Unfortunately, I don't find many followers. I mean, I am one of the ardent followers of this particular concept, that we have to invent a tape. So we proposed a new type of tape. Can you please, David, can you go to the next slide? So what we said that, look, we have to enter, only way we can do that. In 2010, Roger came up with a proposal that if we want to go beyond Turing, I mean, information is integrated in the nature in a very different way. If we want to at all discover this particular part and change the history of humanity and take it to a new direction, then what we have to do is to enter into a cell and then find another world and enter inside another world. So what happens is it becomes undefined. You go within and above, orthogonal to this Turing tape, not side by side. So that's a huge conceptual step forward. And to the left, right, you see my drawing. And this drawing, actually, I drew in 2010. So it's a kind of very historic one. So I was reading this whole thick book, Rogers, and I drew it. Then in 2014, we filed a patent for this kind of machines. Can you go to the next slide? So this is the kind of a situation that we have, that we take one cell, we go inside, and then another cell, we go inside, and journey is endless. So if we can make a situation, then we can integrate information in that particular domain, orthogonal to Turing machine, that cannot be computable in any conventional machine. So we have to make a new type of machine altogether. Even we want to measure any properties of consciousness or any other kind of thing. And that was the biggest challenge for me for the last 16, 17 years in Japan. In our laboratory, we made completely different type of machines. Can we go to the next slide? So the situation we have now, that you have a circle, you zoom on and zoom in or zoom out. you have a journey within and above. And if you want to read it, then you need to measure the phase quantization, how phase is changing, and different worlds, different layers. You need to get into the higher dimensional tensor mathematics, projective algebra, and other kind of difficult things. Because when it is a Turing machine, you walk linearly, everything is so sweet, so cute, so easy. But when it is within and above, it's like third imaginary world interacts with 7 imaginary world and affects the 37th imaginary world. So it is like you are collapsing somewhere which has nothing to do with the universes where you did the information processing. That's the beauty of tensor algebra. I think we can discuss some other time. Can we go? Can we forward? And can you try to play the video this time? Does it work? Okay. So I don't think that any video will be played. So here in the top right corner, you see that there is a blue color atoms. Those are actually water molecules, and orange and yellow color balls. Those are tubulin proteins, and green color these things are C-termini or extended part. So when I first started doing this measurement, and it was beautiful. My postdoc Satyajit Sahu was doing the measurement 2008, 2009, many years back. And when he was doing the measurement, I saw that he was taking big, giant peaks, high power peaks in the kilohertz domain, in the millisecond time domain. And there were very little peaks in the megahertz. And he said, Anirban, those are noise. Okay, can you zoom it? So he zoomed it, and we found in the megahertz domain there are vibrations. So milliseconds, microseconds, and then we found also nanoseconds, entire time domain, beautiful vibration. Just like imagine one tuning fork, you hit it in your hand, you see 1 frequencies. Now 9 frequencies are there, and then all of their phase is quantized. So pi by 4, pi by 4, pi by 4, pi by 4 gap variations. Together, they are an integrated system structure that we cannot write linearly. We need to create a big clock, and we make a phase singularity, and then insert another clock and go inside and inside, just like Roger envisioned in 2010. And we started promoting that as fractal tape, and then we started building a machine. Can you go to the next slide? Okay, so when we have triplet of triplet, and we go within and above, this is a beautiful symmetry, and we in our laboratory we started organic synthesis. So we started creating artificial material, because we understand from the biological system, but if we understand a particular principle, how do we prove that we have understood it? So what we do is make artificial system, and series of patents we filed, and then products we generated. So these are called, chemists call this core cell architecture. So it is triplet of triplet, and then next layer's triplet of triplet is in different time domain, but they have an overlap, so they are coupled. And if you generalize it, and continuously, if you go on different time scale, so you get a total resonance chain. Can you go to the next slide? Okay, so if you do experimental measurement, you find a very beautiful thing. So this is transmission. You take single microtubule and you pump electromagnetic wave, and you find reflection and transmission coefficient. These measurements are available in your university. So you can do this measurement. What beautiful is that, can you see that there is a negative direction, there is also a peak? So microtubule is not only absorbing and transmitting, it is also emitting. So it is not a dead system where you do transistor-like things. So it can triplet, not just triplet of triplet, there are negative things also. So it means it absorbs and transmits also whenever necessary. So it can communicate. It is much more intelligent. So I say the information structure that we learned from microtubule is alive. Can you go to the next slide? Okay, so when we published this paper, and you know that those we are working for ORCWAR or supporting with experiment, we have many friends whose only dream in life is somehow we die. And if I die, I mean, what kind of celebration will they do? I don't know. So when the paper was accepted in Nature, they wrote a letter to the editor not to publish it. So these kind of beautiful people are there. They actually changed my life significantly, because initially I was not that much adamant to orc war, but then I decided that I will finish them off and finish them off in a way that they can never forget in their life. So what I did, I didn't know anything about microtubule or any biophysics. So I started setting up the laboratory, whole laboratory for neuroscience. And this is the first image that you see, that in the axon, we put the coaxial probe, that is atomic sharp needle, because my background was quantum physics, right? I didn't show anything, because today is a friendly discussion between three of us where we are trying to introduce you, and we want to encourage you to come to this field and explore. And in your institute, Professor Dante and Stuart is putting a very hard work along with Tom to create it a center. And there is another reason. Arthur Winfrey in 1960s first came up with this concept in this Arizona itself. So it's a historical place as far as this polyatomic time crystal and everything is concerned. And this Arizona University he worked. And after 2000, it went off and people forgot. So you see that in the neuron also, we found triplet of triplet in a different shifted time domain. So it is not dying down. Which is not dying down? Symmetry. So I showed you in the beginning that somebody simulating neural network in the brain, and somebody is simulating particles in the extragalactic space, and they are following similar kind of symmetries. You see, we are going from single protein to the hundreds of micrometers, 2 nanometer to hundreds of micrometers to millimeter scale. And then we are seeing same symmetry of vibration is followed. Can you go to the next slide? Video will not work. Can you try again? Okay, video will not work. You go to, so this is the tubulin protein. And one of the good things that we did is, I always believed two things in my PhD, that if I ever become a scientist, I will make sure that the palm probe type experiment that is going on in the scientific domain in all over the world, I will just reject it, and I will go continuously opposed to it, because I never felt, I mean, like, palm probe is something like I take a stone, hit somebody, he will scream, and depending on how strongly I hit him and how strongly he screams, I will put a plot, strength of my stone versus screaming sound, and I will get a beautiful plot, fit it, and I will call it the expression of nature. That's the most stupid thing. So I told my PhD supervisor, I will do that. So when I became faculty, first thing I did is I started inventing a machine where we will pump at a, say, very, very, very low frequency or high frequency, and 10 to the power 6 order different frequency domain I observe. Another thing is that I believe that music and dance go together. And without music and dance, you are observing simultaneously. You cannot understand any biological system or anything that is living. So here you see music and dance. So on the top of each image, these are quantum tunneling images. You see with the antenna, we have electromagnetically vibrated those proteins. And we have seen that if the frequencies change, how the conformation is changing. So as if musical beats are changing and you are vibrating in a different way. Can you go to the next slide? And this is also a video. This is also not going on, but you can see that on the surface, the tubulin proteins are there, and at different frequencies, we vibrated the microtubule, also single microtubule, and we observed how electron density distribution is changing. So basically, the different waveforms that is resonating with the microtubule, they are forming beautiful, mosaic on the surface. Can you go to the next slide? Okay, so our journey went on. We didn't stop. We went on exploring many different kind of proteins and similar kind of structures. Can you go to the next? Next slide, please, David. Okay, so here one thing we have to remember, and from today's my presentation, if you learn just one thing, that will be great. That as the time passes by, we could see the kilohertz, megahertz, gigahertz, there is a resonance band and there is a phase shift. But if we bring them together, I mean, in front of my lab, Actually, I put all the frequencies. I never understood there is a triplet of triplet because I was measuring resonance frequencies since my PhD with the spectrum analyzer and all these kind of things. And I could never believe that frequencies are friendly to each other. They are coexisting and they're interconnected by phase. So it took 4 years for me, and suddenly I discovered that actually if I put it on a phase sphere, so microtubule, if you want to understand, see microtubule, then you have to see this 3D spherical assembly. That's the only and the most scientific representation of the information structure. Why? Because each clock is rotating with respect to each other, shifting phase in a very particular manner. Okay, so that not just microtubule, every single living system that will contribute to consciousness should be like that. Can you go to the next slide? Okay, so we wanted to understand, because we are physicists, right? If we don't write Hamiltonian, we don't feel good. So I asked 3 students to come, and then the reason I made is Stuart also. It's very fun, very funny, because I knew that these are capacitors. So I asked just to take the capacitor, solder them, and rotate it. And I hired the students every 3 years, and you make this helices of different diameters and different length, different pitch, because this is the experiment this nature Mother Earth carried out. If you look at the microtubules of all the living species in the planet Earth, you will find the number of protofilament increases from 6 to 19. So she also did an experiment. So we did the same experiment like Mother Earth in our lab and tried to figure out what would be the Hamiltonian. And first student did, this is Karthik. The first student did that, second student did that next year, third student did that. When we found that all 3 created a massive, very large Hamiltonian. I don't want to put fear in your brain, because then you will say we will not do this course or we will not join. So we don't want to do that right now. But these are funny things by which you can understand complex systems. And I will not tell why I took a capacitor and then roll it. And if I do the capacitor and roll it, what would be the implications? Because you got to figure out, students should figure out that why is it. Next slide. Okay, so this video doesn't run, but this video is very useful. What we did is, this is the device, a stupid device playing tools. And if you look at the phase, you will find that 12 different holes on a sphere. And those holes, they blink, they close and open. And that is the information structure of microtubule. The video doesn't run, so we skip it. Can we go to the next? Okay, so you see that we have said that this is the same like Stuart's, that triplet of triplet, and this is almost like a resonance chain. Can you go to the next slide? And we can connect all the components that are there. Can you go to the next? So this is from 2014 papers of ours. Can you go to the next? So can you go to the next? So all the elements were there. So we started measuring frequency, and then we started seeing how lattices change. Can you go to the next slide? So we did the same with DNA and found triplet of triplet. Can you go to the next? So we developed a information theory, and from the resonance frequency shift, we could find out the geometric shape. Because suppose, imagine that there are 3 clocks. Sa re ga, it is equal sides, and sa re ga. So with music, you can feel it. So we called it geometric musical language and build a whole computer in it. The computer runs in our laboratory, and if you want to see www.brainrhythm.org, you can go to the website. We continuously update. We have artificial brain built on that. We will get into that. Can you go to the next slide? Okay, so the Stuart has shown this particular picture, and he explains much better than me. So he has explained this so many times. So I never explain this slide wherever I go and whatever the presentation I give. This is in honor of Stuart, because I thought that nobody in the world would be working on a insulator arranged in a helical shape. So I thought that who would be the greatest stupid like me? And then I found Stuart Hameroff is there. So I went from Japan to Hong Kong to meet him. So the next slide. And we went on finding. So Stuart shows the first slide, but there are many proteins where we try to figure out the triplet of triplet, doublet of doublet, pentet of pentet. What are the kind of inherent mathematics are hidden there? I will not get into the mathematics. Next slide. Okay, so in summary, what we are doing is we see the 2D resonance plot from 1D resonance to 2D resonance to 3D resonance for whole structures. We go. Next slide. And we developed a model for self-operating mathematical universe to explain the orchestration part. So what we do is imagine somebody asks you that you have to create the universe or self-operating system. One of the biggest problem I feel to me in the consciousness research is that no model try to address one thing, that I would or anybody of us are a self-operating system. Forget about consciousness. Consciousness is a big problem. But even before we enter into the world of consciousness, how do I explain this? I had no clue. I brought one presentation and I found that I will not be presenting, somebody else will be changing the slides. So I created another presentation. Then the presentation went there and then none of the videos are working. So I am creating a new presentation in my mind at this moment, and with all the different thoughts putting together and then doing it. Can a computer do this? Cannot. So how am I able to do this? So this is the biggest question. So what I thought is that it's a very crazy idea. Whether it is right or wrong, I don't care. But if I have to find a solution, why? I mean, we should be stupid enough to tell the world that what we think. And maybe it is correct, maybe if it is wrong, it will be rejected. So I thought that we should start from a single point, because if, say, before the creation of the universe, God was not there. So none of the physics principles were there. So what was there should not be there. So that means even if I have a point, I have to add many points, arbitrary number of points into it. Just exactly Rogers mathematics in 2010. So I use that very much, as you can see here in the first line. So if you have arbitrary number of points, the points become undefined. So point even doesn't exist. So I take nothing. So I take arbitrary number of points inside a point, then I take nothing. Then I do calculation that if 3 points, 4 points, 5 points, 6 points, then how can I arrange them? And then I take the probability calculation and found mathematically triplet of triplet. You can go to our websites and others, books and others. You can find out that how we create a triplet of triplet, pure mathematics. Can I go to the next slide? Okay, so then we went on discovering something serious, that we had to organize and create artificial devices. And then a little difficult thing, that is, normally you know that photons or electromagnetic wave goes in a straight line. And if you send the light to a wall, it will be a dot. But it was discovered in 1991, 1992, and I think they got Nobel Prize in 2018, that if you can apply angular momentum, say microtubule surface or protein surface or some structures, then you can add one angular momentum. Then when the light falls on the wall, you will find a ring. Using that ring, you get a lot of information on the surface where it interacted. Now you can perpendicularly, you can add another angular momentum. So here you can see omega, theta, phi, 3 values are there. So using that, you can generate geometric phase, you can generate complex vortex structure. So as you have understood that we are not saying bit qubit, we are going away, much away from the normal conventional belief of information structures, because information structures is the first thing that I said, that we committed a crime, that after the World War II, we didn't throw up the information theory that we created to create slaves or killing machines. We didn't go back to Mother Nature and ask itself politely that what kind of information are you proposing? I'm not saying this is right or this is or everything is wrong, but I think that attempts were not made. If anybody you know, you can let me know. Next slide, please. Okay, so I said that phase quantization is a very important factor. Very important factor. Those who know quantum computing, you know that if a wave goes through a device and then it's out 180 degree out of phase, then you can get C0 gate. And the wave is made of geometric phase, and some device, quantum device, changes its 180 degree, then you get quantum C0 xenon gate. Now see the beauty here. To the right, the 3D plot that I have plotted, you'll find that 0 degree, 45 degree, 90 degree, 135 degree, 180 degree. At all the frequencies, depending on a particular frequency, there is a particular phase. I told you earlier, I'm telling this again, and then even when I leave, I hope that you take this into your heart, that whenever there is a phase quantization of various kinds, so logic gate then it is a little baby. In a quantum logic gate, what do? You take 5 or 6 input and 5 or 6 outputs goes, and then all the output states, you shift them into a particular phase, right? Microtube will not just do that. That shifting also has a temporal function in it. Isn't it beautiful? That not just a gate, it's a combination of gates which operate together as an automaton. That's why I say that information structure is lifelike. So next. Next. Okay, I think that another major mistake that has happened is that I sent consciousness 2 file. I think that has not been sent. It is consciousness 1. Yes. So can you please scroll continuously until I say stop? Because this is not the presentation that I prepared. So these are combinations by which we make phase prime metric. Can you continuously scroll on? This is a wrong file that is running. So we will go to the end and I will draw some conclusion. Can you go to the end? So these are mathematical structures. Can you go? Next. Next. Next.
Might be better just to
No. I requested you that you please make sure that consciousness 2 file is there. So this is the information structure that goes on. And please continue. Please scroll. These are the information structure, information integration process within an above. So you don't do it side by side. And this is a concept of time crystal, how different combinations are created. And those mathematical structure that we say triplet or triplet is basically the reason. And spirals are the structures that you can see in this video. So if the spiral structure device is there, you send light and you create vortices. Can you continue to scroll? Yes, and these are the results we got experimentally from different parts of the biological structures based on the new language. So we want to read and understand nature using a completely different language than before, because we do not want to touch the device. That is the conventional practice. Can you continuously scroll? Yeah, because all these are deleted. And this is a neural network where using microwave frequencies, we can look at the circuits in a very different way. And we can, in ionic motion, if we look at it, circuit looks one kind. And if we look at megahertz or gigahertz frequency, circuit looks very different kind. And go on. Can you play the video? Does this video runs? Okay. Can you go to the next? Yeah, this video runs. Okay. So in 1907, it was believed that membrane is processing information. And after this, in 1982, Stuart came up with the proposal that microtubule is doing the information processing. And 2015 onwards, we started publishing papers that neuron, 2016 first paper came out, that microtubule bundles processes information 250 microseconds before. And it also wirelessly communicates with other neurons and can couple and do the information processing much before. And synaptic firing is required only when it needs to rewire the circuitry. But information processing, it can do directly. But we were not the first group to show that. In 1928, E. Kaz from Germany first showed that if you stop the ionic channel, even then communication occurs and firing happens. Can you go to the next slide? Yeah. So basically, we see that we will have a cavity, and inside the cavity, we will have dipoles or resonators, and they will form a structure, which will be our information structures or information architectures. Can you go to the next? So based on this, we created model for the whole brain. Why I love this? Because Pushpendra Singh, my postdoc did this, and painstakingly, he has gone through every single periodic vibration that happens. I started my presentation using this, and here you see that the whole brain structure has been modeled. All the clocks are there. In some part, there are microtubules, some part neurons, some part hippocampus, and everything. Can you go to the next slide? Can you go to the next slide? Next slide. Yes, can you play the video? Okay, so now you see the brain was like a computer, but now within and above is an architecture of clocks, 3D architecture of clocks. Can you go to the next slide? So we created, when we know that this is a 3D clock assembly, so we created artificial brain, only not to create conscious machine or something like that. We put EEG-like machine on our brain and try to understand that if I have a replica that vibrates exactly like me, what are the new I have non-physical properties that would shed light on the understanding of consciousness. So that is the objective. So several such cool experimental devices we make. Can you go to the next slide? So one of the interesting feature of our work is that 12 time domains in the brain system are useful. So I always say that when 12 time domains or 12 completely independent information structures are generated in a single hardware, then what happens is 2 into 3 into 2. You see this structure generated from one hardware. Can you go to the next? 2 into 2 into 3. Now I combine clocks in a different way. Can you go to the next slide? 3 into 2 into 2. You see the information structures have totally changed. That's why I say that if I want to be conscious in the least possible way, what we can think is I go outside my body, I have another identity, Anirban, that looks at me. But actually that is not true. I need 3 Anirbans to go out. One Anirban is the observer and 2 the others are interacting. And then we can say that this identity is concerned. And then you need a very complicated hardware, as I proposed, that to understand this kind of thing. So can you go to the next? And go to the next. Go to the next. These are many spiral structures in nature. And as you can see that the spiral structures in the galaxies and everywhere. So now I think that will be the best point where I should end my presentation and Dante should take over.
Okay, thank you.
David, can you go back to the first PowerPoint, where Stuart had his slides, after the Q&A, of course?
Okay, so again, as a great deal to absorb, but the students, we should point out that the students, this is the last class of the term, and so the students have broad preparation for a lot of things, and preparation to some extent for this. So if the students have comments or questions, now's the time.
Caitlin?
Hi, can you hear me all right?
Yes.
Yes.
Sorry, hold on 1 second.
What?
I think she's just trying to get it ready. I don't know. Caitlin, are you still there?
I'm still here. Sorry, can you guys hear me now? Can you guys hear me?
Yeah, we
Give
can.
her a
Yes.
thumbs up.
Okay,
Yes. Yes.
sorry.
Only on her there.
Technical difficulties over here. Okay, so based on your research, do you believe that the microtubules are creating the frequencies you have witnessed, or are they being affected by an outside stimulus?
Yeah, so microtubule has water channels and there are many electromagnetic sources, noise. So one study that we did is to identify whether microtubule is absorbing the other signals and then just transmitters, or it can by itself spontaneously generate vibrations. So it can generate spontaneously triplet or triplet this vibration information structures from noise. So what we did is even in the artificial structure that we said with the capacitors and soldering together, if you we sent with a function generator from megahertz to gigahertz noise absolutely electromagnetic noise of various frequencies, and we found a very organized information signal, AC signal generated from the system. So microtubule can generate very constructive signal from thermal and other kind of electromagnetic noise present in the biological system.
Awesome. Thank you so much.
Time for at least one or two more.
Jared.
Hello. Just similarly, I guess, to the question I posed to Stuart earlier is just what would be the form of information that you believe the clocks compute or share?
Yeah, as I demonstrated a 3D sphere vortex, I didn't show the experimental some of the rings, light rings that you saw, and several rings of light. So that is, we say that we are reading the information structure. So information structure is a vortex, is a filled ring of fields or 3D structure. I mean, if you have a water and if you throw a stone, you see rings. So vortices are the information structure in the microtubule, in the neuron. Also, when I showed the video of my neuron, you saw that there are 200 nanometers gap, actin and beta spectrin rings inside the neuron. And you will find that if we do electromagnetic imaging, then you will find 2 rings go. So that means when the ion channel, always in our textbook, you see neuron is a spike, singular spike. That is not true. Action is a spherical cylinder, is a cylindrical shape, right? So all along this circular diameter, all the ion channels should fire simultaneously, release simultaneously. So a ring of fields go there. So this ring of fields is called a vortex. So you can create ring of fields, you can create 2D structure, 3D structure with vortices. So since 1897, vortex is used extensively in physics because they can propagate without dissipation. They can be integrated, they can be used for interference, and you can create a structure of fields which can itself survive and wirelessly transmitted through the system. No need for a circuit. So so many advantages are there. So vortex-based computing, vortex-based many research fields are over there. So we believe that vortex structures are the information structures there. So you can, if you ask that what kind of vortex structures, we don't restrict anything. So there could be mechanical vortex for soliton-like propagations. There could be electrical vortices when you see nerve spikes. There could be electromagnetic vortices. There could be magnetic vortices, depending on what kind of material, what time domain, and what kind of dipoles we are using in the brain.
And you showed entanglement between using vortices. You showed entanglement
Between
in angular
microtubules.
momentum in different microtubules. So there's entanglement in the brain.
So whenever there is a vortex, there is an angular momentum because if the angular momentum is larger, you get bigger ring and a bigger ring.
Can that account for entanglement in the inside neurons in the brain?
Yeah. I mean, you don't have any problem. It has nothing to do with the temperature. All the molecules are inside the molecule. You have quantum properties. If DNA cannot hold its quantum properties in room temperature, your genetic code is gone.
Mhm.
Those are all. I mean, there is a beautiful work, quantum walk. I mean, how proteins understand the RNA code. How do they read it? So that entire process is room temperature quantum walk. So quantum walk is a very unique quantum mechanical process.
So RNA and DNA have triplets of triplets also, but
RNA
they're
and
not
DNA
quantum. They're like instructions or
No.
codes.
That's all the molecular codes in the biological systems are quantum. Molecule cannot hold a classical. I mean, molecule vibration. You need to do DFT, density functional theory. You need to solve the Hamiltonian and find out the wave functions for it. It's a molecule. It's a molecule.
We have time for questions.
From the students, is there one?
Yes.
Petty, go ahead.
So I was wondering if you guys believe in a non-local consciousness, if you guys believe that there can be a local consciousness in addition to a non-local consciousness.
I think I believe.
Yeah, of course.
Yes, I think so. And I think consciousness is actually the transition from quantum to classical. So it involves both. You need the classical to exist in this world, but consciousness per se is a quantum event, is a quantum process, which implies non-locality. So extracellular ESP and all that kind of stuff are possible. It kind of opens a can of worms for classical physics.
Dante,
Okay, before, or you have an answer to this?
No, let's just
you
go.
believe or
Yeah.
not?
Yeah, I think that, no, it's your turn to go,
Oh,
so.
okay,
But
right.
if you want to answer this first.
Do I
that's
believe
okay.
in non-local consciousness? I'm not that advanced yet, so I think my presentation will show you the direction that I'm coming at this from, and then
It's even weirder.
Okay.
we'll see.
Okay, the clock is starting.
Great.
Not
Bring up
three
the
of them,
first
just
slide,
one.
please.
But you can either use
Or
what
here.
I sent you or the first PowerPoint, because it depends on what's more convenient for you.
Yeah, there should be a title slide. And while that's coming up, I'll just give you a little bit of background on how I'm approaching this field. I'm relatively new to it. I think Stuart and I have been talking for about 3 years now. And it was driven by my interest in astrobiology, formation of the solar system, and origin of life. And for the past 20 years, I've been thinking exclusively about sending a robotic spacecraft out to a near-Earth asteroid to collect carbon-rich material from the earliest phases of our solar system, with the hope that there would be indications of how key components got delivered to the surface of the Earth: organic molecules, the water in our oceans, the molecules of our air, everything that makes Earth a habitable world, and everything that is in our biosphere today. Next slide, please. So our target, I'm going to give you a little bit of a background on the mission. I'm not going to spend a lot of time on it because there's a lot of information out there, but we went to a near-Earth object called Asteroid Bennu. The number there is its catalog number, and you can see the orbit of the Earth there in blue and the orbit of the asteroid in orange. It's a near-Earth object, what we call an Apollo type, meaning its orbit crosses directly that of the Earth, giving it a chance of impacting us, which is another part of our scientific investigation I won't go into here today. Next slide, please. You should see the asteroid rotating. This is the object. It's about 500 meters in its longest dimension. That's about the size of the Empire State Building. It's got this really interesting, nearly spherical shape that bulges at the equator from the centrifugal forces. Think of the tilt-a-whirl ride at the carnival. When the thing starts to spin, you're pushed up against the wall, the floor drops out. The same thing's kind of happening at the equator. All that material is getting pushed outward, and it's almost in orbit. This is a very dark asteroid. It reflects about 4% of the sunlight that reflects its surface. That was one of the key reasons that we chose it. That's a pretty rare property in the inner solar system, and we inferred and have since proven that that is related to high abundances of carbon, the key central element to life on Earth. Next chart. This is the site on the asteroid that we selected to collect the sample from. That crater, or that bowl-shaped feature in the center of the animation, is about 20 meters across, about the size of a basketball court or so in the center of the area that we were targeting. You get a sense of the rugged nature of the surface by the large boulders that are surrounding it. And we chose that crater because it had a large amount of relatively fine-grained material that was consistent with our collection strategy. And I'll show you in the next slide how we collected material from the asteroid surface. So this is an animation of the spacecraft descending down to the surface of the asteroid very slowly at 10 centimeters per second. That's a robotic arm with a filter on the end of it. When we contacted the asteroid surface, it flowed away like fluid. This is basically a microgravity environment with no cohesion between the grains. The strategy was, as soon as that filter touched the surface, we opened up some bottles of high pressure gas, kind of like a leaf blower, just blew the gravel and dust through the filter. It was quite a dynamic event, as we captured in this animated reconstruction of the sample collection. We actually penetrated about 50 centimeters deep into the subsurface of the asteroid. Surprisingly, we expected that we would just contact a relatively hard surface and get material from the top. It's good for us because deeper in the asteroid is cooler, and the kinds of materials that we're interested in are more likely to be better preserved. After spending 6 seconds of sample collection, we fired the rocket engines and the spacecraft backed away from the surface of the asteroid with that scientific treasure in hand. That all happened in October of 2020. Next chart. Then we took that filter. This is actual imagery from the spacecraft. It's clicking into a return capsule. The brown part on the bottom we call the back shell. There's a capture ring, which has some cams. It's kind of like clicking your boot into your ski. It clicked in and was nice and secure. So we put it to bed. We tucked it in. Next slide. So we kind of just tugged on it, make sure it was securely fastened. It wasn't going to move during flight. And then we said good night. Next slide. Removed the head from the arm, closed up the capsule. That white nose is a heat shield. So that device, about 80 centimeters in diameter, it's like a mini version of the capsules we bring the astronauts home in. So that heat shield protects the interior from the intense heating from friction during atmospheric passage. The sample stays nice and cool. Kind of like meteorites, we do get these samples from the Earth, except that a meteorite doesn't have that heat shield. It gets directly exposed to the atmosphere. It lands on the surface of the Earth, and these carbon-rich asteroids are very quickly contaminated, colonized, and the bacteria and archaea start to consume the organic molecules, making our attempt to understand if any of the key building blocks of life came from outer space very difficult. And that was the motivation for going out to the asteroid and retrieving our own samples. Keep them clean, keep them pristine, so we have high confidence anything that we find in the sample came from the asteroid and is not contamination from the biosphere. Next slide. Here we can see the capsule being released. So the limb off to the left of the image is the limb of the Earth. The spacecraft is about 60,000 miles away. We spun the capsule just like a quarterback throwing a spiral touchdown pass. That stabilizes its passage through the atmosphere. It's a relatively dumb capsule. That is, it doesn't have any kind of guidance system or active control. So we relied on the entire spacecraft coming back to the Earth, releasing that capsule, targeting a Department of Defense test range in western Utah. And I'm happy to say after we released that capsule, the spacecraft stayed in space. We fired its engines so it didn't follow the capsule into the Earth. It's in orbit around the sun, and it's on its way to another asteroid. And a young assistant professor here at the University of Arizona, Daniela Della Justina, is in charge of that new mission called the OSIRIS Apophis Explorer, or APEX, Apophis being the asteroid that we're going to rendezvous with in 2029. We can't collect a sample or return it because all that hardware is gone, but we have all the cameras and other scientific instruments, so it'll be a whole new mapping campaign. Next slide. And then finally, on that morning of September 24th, 2023, after over 7 years of deep space flight, we got this beautiful footage from our colleagues at the United States Air Force of that parachute coming in for a nice, safe landing, touching down in the Utah Test and Training Range, culminating what for me was a 20-year journey, basically thinking about all of that engineering, all of the challenges, all of the management of making that possible. And as soon as we got the samples in, I got to really start to become a scientist again, and I've been enjoying the subsequent months ever since. Next chart. So here's the picture of the capsule in the Utah desert. It's like the salt flats, the Bonneville salt flats. You might have heard of those. The parachute is sitting right next to it there. Next. I was on the environmental sampling team, so I got to go out to the desert and greet my old friend who'd been away for 7 years, a little charred and blackened from its high temperature passage through the atmosphere, but looking really healthy, safe and secure, no sign of any sample breach. We collected soil, air, dust, vegetation, just in case any of that made its way into the sample. We don't think that's the case, but we have them in case we find something really compelling. We can determine whether it came from Utah or came from the asteroid. Next chart. So we put it into a cradle on a long line and helicoptered it back to a temporary clean room in Utah. Next. The Air Force was waiting on the tarmac the next morning with this wonderful C-17 transport plane. And here we are taking off from Utah, flying that sample to the NASA Johnson Space Center in Houston, Texas. Next chart. where we delivered the science canister into a custom-built clean room. You can see the technicians are already in their clean room gear. Normally, you wouldn't wear your clean suits outside of the clean room, but since we were transporting it in, it was one of the few occasions where we allowed that break. And next slide. We got that canister opened up. We see the sample collector safely still secured in its capture ring. Everything looked nominal. Next chart. And you had a big happy principal investigator because for the first time in my life, I actually got to lay eyes on this asteroid that I've been dreaming about since 2005. And it is dark. It's an amazing experience to see material that only reflects 4% sunlight. It's darker than coal. It's darker than asphalt. It's darker than anything I'd ever seen before. It looked like holes in the deck, even though I knew they were grains of material. When I show you the images of the sample, we do stretch them so you can see some contrast. Next chart. This was the first site that we saw as soon as we got that canister open. There's the sample collector, very much that air filter design. But what was exciting was there's that ring of black dust and then kind of a dashed pattern on the outer edges there. Those are all small particles that were pushed to the edge of the canister because we secured it with a nitrogen purge, nitrogen being the most abundant gas in our atmosphere and generally chemically inert. So it's easy to produce and it minimizes any risk of contamination. So we don't let the sample see air. We don't want it reacting with oxygen. We don't want it reacting with water or anything else in the atmosphere. But we accept that we're going to have to expose it to nitrogen out of practicality. And it is the most convenient way to keep the sample relatively pristine while it's resident on Earth. Next. And then here is the majority of the collection. This is about 70 grams, a little over 2.5 ounces of material. The requirement was 60 grams or 2 ounces. We ended up returning 121.6 grams or over 4 ounces. So we got more than twice what we promised. The largest stone is in the lower right corner there. That's about 3.5 centimeters, and it's pretty flat and flaky. We've got about a dozen particles on the scale of a centimeter and thousands of particles at the scale of a millimeter. This is a fantastic collection that will be studied by research for decades into the future, just like we're still studying the Apollo lunar samples that were returned in the late 1960s and early 1970s. Next. So that's a 10-minute whirlwind through 20 years of activity. There's a lot of documentation on the mission. I encourage you to go on the internet. I have a TED Talk that just got posted all about it as well. But now we get to the real reason that we flew the mission and the scientific question that has driven my career, that drove us to get out to that asteroid, which is to really understand this amazing transition that I kind of show on the cartoon diagram over there on the right, which is that you somehow start out with those rocks. They were delivered, the material like that was delivered to the surface of the early Earth after the asteroids were impacting large enough to spall off the moon. There was a complete magma ocean on the surface of our planet. That all cooled down, and carbon-rich asteroids like Bennu probably landed on the surface of the planet and brought some of these basic molecules. For example, water, ammonia, which is the NH3, methane and other hydrocarbons, the CH4, hydrogen sulfide, and phosphate. Those should look pretty familiar to anybody that studies biology. Those are the most abundant elements in all living organisms on Earth. Then you go up the chain. So you start out with rocky material. It's got some of these volatiles. That's what we call those compounds that are stable, mostly in the gas phase, even to low temperatures, and then condense as ices. And you move up the block and you get to what we call prebiotic molecules. So not simple water, ammonia, methane, but molecules that look like the things we use in biology, like the nucleobases, which are the letters of our genetic code, and the amino acids, which are the building blocks of our proteins, and the lipids, which come together to make watertight barriers or cell membranes. You got to move up another level. You have to make the nucleic acids. So you take the nucleobases, you add a sugar and a phosphate, and then they can chain and link for thousands and thousands and thousands of components, building up our genetic library, which is basically a useful way for the cell to have access to lots of different proteins that it might need for its function. The amino acids polymerize through the peptide bond to form protein chains, and the membranes encapsulate all of that, and then somehow that becomes alive. You have all of those systems, all of the complicated processes like Anirban's been showing us, Stuart's been showing us. You make this transition from a non-living system of chemicals into something that we would clearly say is alive. We struggle in astrobiology with the same questions. What is life? And now we're bringing to it, what is consciousness? And as I was wrestling with this, really starting in late 2020 after we collected the sample, I just had this feeling that maybe feelings were involved and that consciousness, the fact that I was asking the question maybe was a hint to how it all began. And I started going online, going to journal articles and seeing if there's anything like what I'm thinking about. And I found Stuart's paper on the quantum origin of life. And I really loved it. I resonated with it, I would say. And I spent days and days reading through it. I was reading other theories of consciousness as well, like integrated information theory. But what I liked about Stuart's was it took it all the way down to the molecular level, which is the level I was thinking at, the level that I was working at. And it had very specific testable predictions that I could use my sample for. And that to me made it a natural for me to reach out, and fortunately made a good friend. And we've been thinking and working together, still learning from Stuart, and now working with Anirban, learning an enormous amount. Even just in the past couple of weeks, my understanding has grown enormously through our conversations and interactions. So this is the big puzzle. How do you go from basic molecules, which we know are abundant in the universe? All of those are in the sample. I won't go through all the details, but the sample is very water rich. It's got a lot of ammonia. It's got hydrocarbons like the polycyclic aromatic hydrocarbons, sulfides and phosphates. And as we'll see, even some of these prebiotic compounds. Next. So Stuart and I have put forward this, what we call a bold new idea, is the consciousness first origin of life, which says that consciousness preceded life and provided the motivation for molecular self-organization. I just presented this last week at the Science of Consciousness conference, and some of my charts are in response to the feedback that I got there. So if we go to the next one, it was really, what do you mean by consciousness first? So I said, okay, I need to explain it for the audience, and I need to explain it for myself because it's not always obvious what we're talking about. And I went to two of the presentations that we saw there and that I thought gave a good description of what I mean by consciousness first, and I think what Stuart means as well. The first one was by Hartmut Neven. He's a vice president at Google in charge of their quantum AI, so the team that's building quantum computers. And he says consciousness is how a system experiences the emergence of a unique classical reality. And so the key here is, what is the system? And for me, the system is going to be maybe the protocell or the first molecules. And somehow, if I'm looking for a consciousness first origin of life, I'm looking for a molecular system that can get set up into a quantum superposition and then experience the emergence of a classical reality. So you're looking for something that can be quantum and then make that conversion, and it responds to it in a way that we would think is lifelike. The other one I liked was from Santosh Helekar, who's at the Weill Cornell Medical College. And he said, consciousness involves modulated, spontaneous conversion of waves into particles or collapsed decoherence of the wave functions of electrons, protons, or ions in the brain. So Santosh's definition is very much brain-oriented. I don't think that's necessarily true. I think you could take away in the brain and say in the primordial soup, and maybe we're talking about the origin of life there. And so what do I assume? I'm not evaluating different theories of consciousness. I'm taking the orchestrated objective reduction theory and saying it's testable, and that we can test it for the origin of consciousness and see if that tells us something about the origin of life. We've heard a lot about how it's been proposed by Roger and Stuart, and that it flips the measurement problem, the classic Copenhagen interpretation of quantum mechanics, that a measurement collapses the wave function. Some researchers took that even farther. It has to go beyond measurement, and the measurement has to register in a conscious system. That would be von Neumann and Wigner. And it flips it. It says when that collapse occurs, that is the conscious moment. So it kind of runs it in backwards. And that basically we're looking for quantum computations in humans and other advanced life forms in microtubule proteins to regulate neuronal firings and mediate consciousness. And we're looking for something like that in prebiotic compounds. So the basic strategy is they're putting together a very compelling experimental set of observations in support of these quantum processes and microtubules and their orchestration. But I had to go and retell the story to make sure I understood it, so you can grade me on the next set of slides, which is my understanding of the orchestrated objective reduction hypothesis. Next. And I was playing with my AI image generation as I was going through this. So we got some funky images here too. So the way I'm thinking about it is we're stretching space-time. Imagine space-time is this stretchy rubber sheet. And if there's nothing else in the universe except space-time, it's just going to be flat. But Einstein, classic theory of general relativity, says that mass is a stretching of this sheet of space-time. And so this is what gravity is. Gravity is that well that's created by the mass, and if you get the right velocity, you can orbit around it forever. And I kind of had it drawn as these little tracks that the planets orbiting around the star are following there. So that's very well established. It's been probably the most experimentally verified theory in all of human history. General relativity has passed every test that's been thrown at it. Next. And there's been many. And so then you have collapse into classical states. So in orchestrated objective reduction, instead of planets or stars, you have the stretching occurring at these really tiny quantum scales. And when you have a system in quantum superposition, which is also very well established experimentally, the system evolves into separate worlds as the wave function propagates through time. But that's not a stable configuration because you're stretching that rubber sheet. So I think of this like a rubber band. You can pull it, you can pull it, you can pull it until you can't pull it anymore, and eventually it's going to snap back into its relaxed state. That's the collapse of the multiverse into the single classical reality. And that snap of the rubber band is the Bing, is Stuart's Bing moment. And that's a moment of objective reduction. There was no observer who caused the collapse. It was simply the self-gravity of a multiverse stretching to the point where it can't stretch anymore. And that is where quantum information is translated into classical information. And maybe Anirban can help me understand if I got that right too. Next. Okay, so how does this link to consciousness? So I think of this as like a network of trampolines now. They're all connected to each other, and each trampoline is a microtubule in a neuron, and it has not just one mode, but many, many modes that you could be bouncing different balls on. And that when a ball, which is our quantum information, rolls onto the trampoline, it stretches it down, and at random, one snaps back, propelling it into the air where it can influence the position of balls on nearby trampolines, and each snap back is a moment of consciousness, and the quantum information held in superposition collapses to a specific state, resulting in your isolated conscious experience. And so finally, you have to orchestrate all of this. Next chart. The orchestration. So now it's like a symphony, which is the brain's network of neurons. Each musician or neuron is playing the strings of their microtubules from a sheet of quantum music that could result in many different melodies, which are all the possible quantum states. However, when the conductor, which is a mechanism in the brain, potentially influenced by fundamental space-time, like we talked about, decides, the orchestra suddenly shifts from playing multiple possible melodies to one single song, which is the classical state. And this sudden shift in the music represents individual moments of quantum computation within the neurons, which contribute to a unified classical experience of consciousness. So each snap or collapse is like the conductor deciding the next note of the symphony, tuning potentially into experience. And I really like the ChatGPT image there of each musician is a neuron playing a microtubule, and then it gives you the orchestrated brain up there. So these AIs are really fun. Next. Okay, so where are we now in the science? There's a lot of different areas. I'm not going to go into all of them, but one area that I think where I've tested an idea that came out of the discussions with Stuart, and it's been validated, is this change in the fundamental structure of cells that happened right around the time oxygen rose in the atmosphere. Photosynthesis was invented about 2 billion years ago. The surface was fully saturated and oxygen started to build up in the air, and that was a poison. Oxygen makes things burn, and the cells had to respond to that. So you went from the very simple kind of cell, which still exists in the bacteria and the archaea today, shown by our favorite E. coli prokaryote in the lower right part of the diagram, compared to a neuron, which has a cell nucleus where the genetic library is stored. And then you can see all the green tubes are the microtubules. And we don't talk about it a lot, but there's the centriole, which to me seems like the conductor of the whole thing. It's this very tight arrangement of microtubules, and it does a lot of work in other cells. Next slide. So we've heard a lot about this, the quantum processes in proteins. There is a neuron. It's got the microtubules running down it. They're made of the tubulin dimers, which are the final diagram over there on the right. And then the colors correspond to the different aromatic amino acids. As Stuart said, tubulin is characterized by having the highest concentration of these aromatic amino acids of all the proteins that we know about. That has to be a reason for that. There has to be a reason that the neurons would concentrate those particular chemicals. And that's where we come in, because we're looking for those molecules, those amino acids, phenylalanine, tyrosine, and tryptophan in the sample. Next. And why do we care about tryptophan? Well, tubulin contains a large amount of this. It is known for its large electron clouds, and it's characterized by the double ring there, the indole ring. And it is critical in the microtubules because its electron clouds, and I would like to hear Anirban, if I'm getting this right, contribute to quantum coherence and quantum entanglement within the tubulin proteins. And the electron clouds of tryptophan could help form conductive pathways along the microtubules, potentially supporting quantum computations that ORCO-R postulates. These pathways could facilitate coherent oscillations and transitions between quantum state across the microtubule network. And the quantum states influenced by the placement and properties of tryptophan might collapse due to the gravitational effects related to the space-time geometry as proposed by Penrose. So it seems like tryptophan is central to the rich conscious experience that some humans enjoy. Next. So if we take a look at what do we have in the sample, here are all the 20 amino acids that are used biologically. There's 21 here, actually, because there's a rare one that shows up sometimes. Next chart shows the ones we found in the sample. Next, yep. So anything that's outlined in green has been found in the asteroid. If it's got a dashed line, the tyrosine is the only one that's dashed. That's tentative. It's right out of detection limits. We're just released some more samples, so we'll clear that out. And the ones with the red strike through have not been detected. So far, 13 of the 20 amino acids that are used in biology are coming from this asteroid. There might be a 14th, and then there's 7 that are missing right now, one of which is tryptophan. And that's really interesting if we go to the next slide. So if we look at the microtubule, we have the phenylalanine, we might have the tyrosine, but we're missing the tryptophan, which would imply early life might not have had access to that more advanced molecule. So the next chart. This just shows you what the centrioles and the microtubules do during mitosis or cell division. So they are kind of the workers or the orchestrators of cellular processes for cells that are still dividing. And what's interesting about neurons, once you reach a certain state in your development, you don't make any more neurons. What you have is what you get for the rest of your life. So they go from this active function of cell division to playing a separate role in neurons with information processing and coordinating quantum state collapse. But to me, this is an important clue of where the microtubules came from originally. So if we go to the next slide, we look at what is doing this in the bacteria that don't have the microtubules. They have a different protein called FTSZ, and this is an evolutionary ancestor of tubulin. And it does the work of cell division in bacteria. And you can see it's tagged fluorescently in the little movie on the upper left there. And it forms a ring, and it goes to the center of the cell, and it squeezes the cell walls until you have 2 new bacteria. And they show a lot of similarities, FTSZ and tubulin. However, there is absolutely no tryptophan in the filamentary proteins responsible for cell division in the prokaryote cells. And it's substantially enriched in phenylalanine. So this is consistent with our asteroid data. The asteroids look like they might have been delivering phenylalanine. Tryptophan is a much more complicated molecule. It's metabolically the most expensive amino acid to produce. We can't make it ourselves. It's an essential amino acid. We have to eat it from an organism that can make it. So the story that's emerging is that asteroids delivered some of the aromatic amino acids, the simpler aromatic amino acids, ones that had just the six-sided benzene ring. As life originated, I think this protein has to be pretty ancient because it's playing a central role in life, which is reproduction. If you don't have something doing the function of separating the cells, you're not going to make 2 bacteria from 1, and then 4, and then 8, and then 16, and get the exponential growth that they're famous for. And you've got to remember that process has been going on since the origin of life. There's been reproduction and new cells formed. Basically, it's an infinite process as long as the resources in the environment are available and stable. So next. I just want to end with where we're going with this research. I'm really excited about the collaboration with Stuart and especially with Anirban and all of his analytical capabilities, as well as all of his computational capabilities. So in the upper left there, we've got a macromolecule. That's not from the Bennu sample. That's been isolated from one of those carbon-rich meteorites. We are going to be looking for things like that in Bennu, and I fully expect we'll see them there. The first thing Anirban did was break them off into all these interesting functional groups, which are well known to the pharmaceutical industry. And then he was able to measure this, to model what the structure would be in 3 dimensions, showing that it's kind of got a helical form to it. And then all the way over there on the right, that is the propagation of a magnetic field. And so this molecule already can take one of those fields and put it into this cyclical pattern, very similar to what Anirban is showing in those microtubules. So even these primordial large organic molecules from space are showing some of the fundamental properties and interactions with electromagnetic radiation that Anirban's been demonstrating in the microtubules. So the strategy is, I think the theory is getting very well supported experimentally. A lot of that experimental evidence is coming out of Anirban's lab. So we're getting a pretty good sense of the nature of quantum information and information processing in the microtubules. And if we can see some similarity in molecular behaviors in these primordial compounds that formed in the early solar system, I think we'll start to gain experimental evidence for this consciousness first idea, consciousness being defined very pragmatically, like I did in the middle of the presentation there, a system experiencing the emergence of a classical reality from the collapse of quantum uncertainty. And I think that's my last slide. Next. Yes, thank you very much.
Okay, while Dante projected himself as a student, I think we should let the actual students in the class ask some questions as well.
Jared?
Thank you. So, it's been proposed that tubulin is kind of a computationally capable biomolecule, and what I was seeing of like the magnetic field kind of resonance or structure that you showed of that other larger organic molecule that's been, then it might, like other or macromolecules like that, have computational capability.
I think that's an Anirban question. So do we think the macromolecule that we found in the meteorite might have computational capabilities?
Yes, but there is one thing. It is a Pandora's box, because the science that we are going to see in this kind of molecules, because tubulin protein is enormously complex and it has many different kind of alpha helices, secondary structures, and something much more complex. But in this case, very cleverly we see that the polyaromatic hydrocarbons are replacing some of the beta sheets and taking over and playing the roles. So the science that is there, they look similar, but the science is much more sophisticated and advanced.
Of course.
Yes.
Yeah,
So
the evolution has optimized the tubulin for information processing and conscious experiences. Right? And these are, we don't know what was the motivation for the formation of these
exactly.
molecules.
So there is a lot to learn about why and how nature did this, and especially why.
Right.
Because because there are observation of triplet of triplet because of the helical structures, and exactly that we said it was observed in the meteorite derived this molecular structures, but the way it was generating the result is very different, and our understanding is not yet clear that why.
Right.
In the quantum mechanical calculations, DFT calculations, and electromagnetic field simulations, how it will distribute and all these kind of things are showing unique nature. But I feel like a completely new type of biophysics was playing out there.
Yeah.
And
I think the students should appreciate this is about 2 weeks old, these ideas. I mean, we've just really started putting our heads together, the three of us. And already amazing things are coming out, enough to get me excited and encouraged that we're going to have some breakthroughs, but it's very nascent. The sample analysis is very early in the program. We're just starting to get large masses of material to the team, which is on the order of grams. We've been working in milligrams just to understand the material. And then this synergy and this collaboration is just taking off. So there's a lot of work ahead of us.
Okay.
Do you guys, sorry, do you guys think that there's a chance that we can't measure some aspects of consciousness? Like, it can't be empirically proven that it's like, I don't know, like something we can't measure. I don't know how to explain it other than in those words.
Yeah, I think it's a common perception, and science. Part of the problem is that this community has been, especially with Stuart at the vanguard, has been pushing to make it a realm of scientific inquiry, because that was kind of the standard response. It's not something amenable to scientific investigation, but it's clearly a phenomena in the universe. It's challenging because we've tried to make all of our physical sciences objective with the human observer outside of the system and not influencing it. But quantum mechanics blows all that away, right? I mean, quantum mechanics is where consciousness was forced into our universe, the scientific universe, and we've been grappling with that ever since, I think, as a species.
I would say that what we can measure is if a process is inhibited by anesthesia. Anesthesia is selective for consciousness. In patients who are under anesthesia, their brains are still quite active. The only thing that's missing, it seems, is consciousness. And it's probably because consciousness is a quantum effect, and anesthesia acts by quantum interactions and only affect quantum processes. So you might, as Christoph Koch criticized me once for suggesting that anesthesia could help explain consciousness. He said, well, anesthesia is too nonspecific. It binds all over the place. It's in fat stores. It's in all over the body, and it's very nonspecific. And yet, I pointed out, and it's also selective. And I think the answer is that it only affects quantum processes that are highly orchestrated. Quantum processes all over the body are random, and so they're already randomized. So anesthesia comes along and randomizes, doesn't do anything because they're already random. It only affects highly orchestrated quantum processes, which are consciousness, and that includes microtubules primarily. So any of these little processes that we've seen, whether it's the simulation that we showed from this molecule from the Murchison meteorite, or when we get the samples that can start doing quantum optical effects and seeing things like the magnetic field fluctuation through that molecule, then we can try to anesthetize it. And if it goes away with anesthesia, I think we can make the argument that that might be relevant to consciousness and support the idea that consciousness was there from the get-go.
Another question from Jared.
Thank you. Just to piggyback off of Faye a bit, on how the experience of consciousness still being, to quote David Chalmers again, still the hard problem. Could it be a case in reality that like the computational underpinnings of that generate consciousness might be a completely different problem from our subjective experience, or how that happens?
I think you can have consciousness without computation. I mean, if you meditate and you achieve samadhi or pure bliss of nothingness, there's not a whole lot of information there. It's just pure feelings. And you know, when people try to use complexity to explain consciousness, I say, well, what's complex about a toothache? So I don't think I differ with some of my friends about this, including Dante to some extent, but I don't think complexity is that, or even information processing is the key. You need something to be cognitive, but you could have experience without information processing, I suspect.
Information helps get rid of the toothache.
It tells you what to do about it,
Right.
but it doesn't really get rid of it.
Any other questions there?
Oh,
Is there any reason to believe outside of two humans like touching near that there would be interaction between brain to brain, like at a distance?
It's possible if consciousness is a quantum effect. So our theory differs from the standard classical theories because they say no and they won't go there. They rule out a priori ESP, true out of body experiences or afterlife. And I don't say that those are true, but I think we cannot exclude them until we know what consciousness is in the brain. We can't exclude it out of the brain. So I'm open to that, but I don't advocate it because I got enough problems.
Right. But quantum entanglement allows for it.
Sure.
You can absolutely have systems that are entangled and they can be separated by thousands of miles, probably even more.
Right. And more and more we're hearing about brains synchronizing, whether it's human brains with artificial brains like Anirban's or just human EEG and training if they're doing something sympathetic in some way. So I think non-locality has become more and more, both in space and time. I think there's actually pretty good evidence for backward time effects in certain processes. Otherwise, we couldn't have free will. We'd be because if two people are talking and somebody says something and you look in the other person's brain, and that person will have already responded by the time the activity occurs in the brain, and therefore it's asserted that we respond non-consciously with our non-conscious autopilot and have a false illusion after the fact that we were conscious. And with backward time as effects as shown by Benjamin Libet and as allowable in physics according to Roger, then it could be that we have this backward time effect that allows us to actually act in real time and have free will.
Thank you.
Jared, did you have another question?
Yeah, you mentioned Anirban's artificial brains, and I wanted to ask a few things about those. They seem really cool. Just like, to what degree of complexity are your artificial brains similar to actual brains? Like, do you have it down to complete neural networks replicative to actual brains?
So neural network wise, very accurate, because from 1 millimeter to upwards, we used 3D printing to exactly replicate the structures that we have. 47 different kind of organic gels were used, which were tested to the molecular scale, and below 1 millimeter, we started from single molecule and self-assembled it to 1 millimeter resolution. So neural network were exact replica of all, and throughout the brain, others. Couple of things that we artificially induced. For an example, blood vessels vibrate our brain very, very significantly. You know that every time our heart beats, our brain boom, boom, boom. It's a big blow that goes over there. So we simulated that with ultrasound vibrations because we didn't put the blood flow. Instead, we had fibers that were vibrating, and so as far as neural network is concerned, exactly as is. And the clock architecture that we saw, that was compilation of two things. First is our study on microtubule neuron and some of the proteins, but we are lucky that we are born in this time. Last 100 years or 130 years, researchers have documented every single clock in the brain systems very, very accurately. So those are in the literature database. So we downloaded those databases and then we reconstructed the whole structure. Many of the structures we are also thankful to the Connectome project, which sliced the brain and from 250 micrometer resolution, they created those structures. But those structures are very low category, low quality, and even if you pay money to buy them, you can't use it for the study that we did with electromagnetic resonance. So you can go to our website and download it free, or contact us, we give it free, because we reconstructed the whole structure. The problem is when you slice the brain and you map it, then there are errors, and one neural nerve fiber and other nerve fibers, they get virtual connections which does not exist in reality. So we went through each and every fibers and then we deleted those and then recreated the brain's fibrous network. So you can download it. So we have painstakingly created the whole brain structure database, which anybody can download. It's free. I said in my talk, www.brainrhythm.org. Currently, we have it in our laboratory, and if you come, then we put one DDG machine dodecanogram. As I said, brains have 12 different time bands. So we put on artificial brain and your brain, and you can converse with it, and we see how are the differences are happening. We carried out on 289 human subjects, and we identified 97 perception, cognitive and other kind of things. What are the clock geometries would be, that the geometric invariance for each cognitive functions for humans that should be independent of race, creed and religion and other kind of identities. So all these are documented in our new book, Mysteries of the Brain, I think from Springer Nature. The book is out in this March. So you can go through our entire journey of human subject studies, how with using artificial brain, we did it. So just imagine that we are not doing it for understanding or creating robot's brain. The primary objective is that I believe strongly that there is a non-physical entity associated with consciousness. And if I can make artificial brain vibrating in the exactly same like the human brain body network system. So our human brain, if you look at it, we have a hands nerve network also, because for me symmetry is important. So two legs, two hands, and then the body, spinal cord, everything is part of our brain. So we do not consider brain that is inside the skull here. The spinal cord is an essential part, and all the nerves. So everything we replicated, and we put it inside the jelly, inside the liquid, and with fat. So instead, in the beginning of our presentation, I showed that actual brain is 62% water, then 12%, 16%, 60% proteins and oil. And if you put and mix, you get it. So in our case, it is 16 plus 62, it is 78% oil. So pine oil and other kind of oil. So we have checked the electromagnetic resonance, how vibrations could be replicated, complex set of vibrations, this triplet of triplet structures. And we took oil instead of water-based, is because oil will give us a longer survival. If it is water-based, we need to, because we are water-based, right? We need to regenerate the structures and degradation process. You know, every moment we are here for 1.5 hours, all of us, and including all the students present there, have created zillions and zillions of protein molecules synthesized and replaced the old ones. So we have to go through those processes. So we bypass those kind of things. So there are many weaknesses that we had to bypass, but in majority, I don't think anywhere in the world, even to this level, anybody even attempted to build artificial brain. It's organic.
Okay, I
You're
think-
not claiming it's conscious, though.
No, no, never. I don't want also.
I think that the hour has arrived. So I would just close things for the students. Many of you, given the modern world, are going to be around for 50, 60, 70 years, maybe more. When you see the headline in the Science Times that the problem of consciousness has been solved, and you notice that it resonates with what you've heard today, you can remember that you were there at a certain stage, at any rate, in the development of an understanding of what conscious is and how to study it and perhaps expand it or do other things with it the more we understand about it. That's the end of this class. Thank you to the three experts. I don't know which of you played the role of Copernicus, which Galileo, and which Newton. I would say that each of you have a little bit of each in terms of your contributions. But the three of you, you'll pardon the expression,
Triplets.
make a whole that is fascinating and as the science of all of this develops.
Thank you, Tom.
Yeah, thank you. Thanks
you,
to
Tom.
the students.
Thanks, everybody, for
And
watching.
thank you to the students.